The spike protein is one of four major structural proteins present in COVID-19, and is responsible for the binding of the virus to ACE2 proteins present on the surface of several human cells. It's composed of three identical protein chains, forming a trimer. The cryo-EM structure of the spike protein was first elucidated in its pre-fusion conformation, with the receptor binding domain not exposed, as seen here. The trimer structure of this protein shows many similarities with the SARS-CoV spike protein, sharing 98% sequence identity. The receptor binding domain, present on the S1 subunit, is responsible for viral attachment, prior to fusion and cell entry carried out by the S2 subunit. It has been reported that the receptor binding domain of COVID-19 has an ACE2 affinity around 10 to 20 times greater than that of the SARS-CoV receptor binding domain, with other studies observing similar affinities. Tai et al. Found that a recombinant SARS-CoV-2 receptor binding domain protein is capable of inhibiting SARS-CoV-2 attachment to ACE2, confirming its function. The receptor binding domain in the down conformation is incapable of binding ACE2. Therefore, receptor binding domain extension must occur, as seen here. In the extended conformation, the chains present in the trimer make 1,400 angstrom squared less contact with one another. This reduced contact results in increased flexibility of S1, containing the receptor binding domain. The increased flexibility of the spike protein, observed here, is the main reason for the increased ACE2 affinity seen for COVID-19. With the spike now extended, interaction with ACE2 can be initiated. The receptor binding domain and ACE2 contribute 17 and 20 residues for interaction, respectively. 8 out of the 17 residues contributed by SARS-CoV-2's receptor binding domain are shared with that of SARS-CoV. Interactions between the COVID-19 receptor binding domain and ACE2, seen here as dotted yellow lines, have been reported at 2.45 angstroms. Many attempts are made by our bodies to eliminate the threat of COVID-19 infection. Antibodies are essential components of the viral defense mechanism in humans, and serve as lifelong protection against viruses once attained. Human neutralizing antibodies target the ACE2 receptor binding domain of the spike protein, in both up and down conformations. Here, you can see the Fab domains of the COO2 human antibody, binding to the receptor binding domain of the spike protein. Once bound, the spike protein can no longer interact with residues of the ACE2 receptor, therefore preventing the initial stages of COVID-19 infection. This viral defense mechanism has inspired the production of nanobody inhalation treatments for prevention of COVID-19 infection.